Our next speaker is Johannes Linder. Johannes, if you please come down here. Yeah, I'm set on your computer. Johannes, as Jesse was mentioning, did his postdoc studies with Anshul Kundaji at Stanford and now works as a machine learning scientist at Calico Life Sciences with David Kelly and several others. And so we are very pleased to have you here today, Johannes. And your title is Predicting Gene Regulatory Function from DNA Sequence with Deep Learning.
Great. Thank you for that introduction, and thank you for the organizers for inviting me today to give a talk. Let's see, I'll be using this one. Great. Okay, so this is my disclosure. And so today I'll be sharing some of our work in David Kelly's group, where we develop sort of these machine learning models that allow us to predict various types of gene regulatory function from DNA sequence. And sort of what we aim to do with these models is to functionally interpret genetic variation to aid us in interpretation of various variant data sets. So as we all know, the human genome consists of these various regulatory layers, ranging from chromatin accessibility to transcriptional activation to post-transcriptional processing like isoform processing and other regulatory steps. And in Dave's group, we developed these parametric machine learning models that take the genomic sequence as input and are then trained on various data sets to predict these regulatory functions from sequence. And there are many interesting and important downstream applications for these kinds of models. In Jesse's excellent talk, one really cool application is to actually de novo design or edit regulatory sequences. The application I'll be focusing on today is to actually use these models to functionally interpret genetic variation. So as a brief overview, I'll start by describing a neural network model that we developed called Borsoi that allows us to predict raw RNA-seq coverage patterns from DNA sequence. And then hopefully I'll have time to highlight some of our most recent work where we extend this modeling framework to actually predict pseudobulk cell type specific single cell RNA-seq coverage patterns and sort of adding a cell type specific spin to the model. So beginning with Borsoi, and as a general introduction to the problem that we're trying to tackle here, the human genome consists of more than 3 billion base pairs. The vast majority of these nucleotides do not actually code for a particular protein. Instead, these nucleotides make up a complex sequence regulatory code that governs gene expression through multiple layers of regulation. For example, at the layer of chromatin, we have enhancer and repressor elements spread out throughout the genome that control the transcriptional activity of genes that can be very far away. Then, as a gene is transcribed into pre-mRNA, it's further subjected to additional regulatory processes, including splicing and polyadenylation. And to some extent, these processes are themselves regulated by a sequence code embedded in the pre-mRNA molecule. Finally, this now mature mRNA transcript can go on and become protein product. And along that journey, it's again further subjected to these various regulatory steps, such as mRNA degradation or control mediated by translation efficiency or other mechanisms. And these mechanisms are themselves controlled by a sequence code, at least to some extent, that in this case now is embedded in the mature mRNA transcript. And it's well known by now that numerous non-coding genetic variants are implicated in genetic disease by disrupting these various regulatory functions. But to this day, it remains extremely complicated to take any given genetic non-coding mutation and understand at a sufficiently detailed level how that mutation is potentially impacting each of these regulatory functions. And to make things more complicated, these mutations may have cell and tissue-specific effects. So in Dave's lab, we're trying to approach this by training these high-capacity machine learning models on large-scale molecular data in order to predict these regulatory steps from sequence alone. And with those kinds of models, we can then use them as kind of simulation engines to simulate the impact of any individual mutation in isolation. And really, by applying interpretation techniques to these trained models and to their predictions, we can try to elucidate what in the underlying sequence grammar was actually disrupted by that mutation. And as a brief motivation for why we're interested in doing this at Calico Life Sciences, we're very interested in studying the hallmarks and mechanisms of aging and aging-related disease. And one powerful way to do that is to study genome-wide association studies, as many other people in this room do. But as we all know, it can be extremely complicated because you can get hundreds to thousands of significant hits, and these SNPs can be confounded by linkage disequilibrium, meaning that it's very hard to tear apart the actual causal variants, not to mention actually functionally understanding how those mechanisms are acting. And so that's where we're hoping these machine learning models can come in and help us prioritize variants that are more likely to be causal, and by giving us a grounded interpretation of what in the sequence was disturbed. So one rather flexible and quite powerful approach for modeling many of these regulatory functions is to reduce the problem to that of learning to predict genome-wide sequencing coverage assays as kind of one-dimensional coverage profiles, given only the genomic sequence as input. And this approach was popularized by the Informer model that was developed by DeepMind and by my PI, David Kelly, at Calico. Recently in Dave's lab, we developed this new model called Borsoi that takes that idea kind of one step further, where we're now teaching the model to actually predict raw RNA-seq coverage patterns in a tissue-specific manner from sequence alone. The intuition here is that if a parametric model can learn to predict RNA-seq coverage patterns, over the exons of a gene, it must have implicitly learned about many different regulatory steps sort of in one single model, right? It must have learned about transcriptional activation and chromatin accessibility. It must have learned about, splicing and polynylation in order to identify the exons and the relative magnitudes. So we think that here you're learning about many regulatory layers in sort of one go. So architecturally speaking, the model borrows from many different ideas in the ML literature, but of course builds on many of the successful components from the predecessor Informer model. So to start off with, the Borsoi model is shaped as kind of like a unit, basically. It takes in this huge chunk of genomic sequence, roughly half a megabase. The sequence is then transformed through a number of convolutional layers interspersed with subsampling layers. The convolutional layers are sort of learning different salient regulatory motifs, and the subsampling layers are reducing the sequence length into a manageable length such that we can then apply these self-attention blocks that allow us to quite efficiently transmit long-range information from one position to every other position in just sort of one layer of computation. And then finally, to actually predict RNA-seq coverage, we want to do that at a reasonably fine resolution, because some exons can actually be quite thin. So then we borrow these the approach of unit layers from the computer vision literature, where we're now upsampling the signal again with convolutional layers and these kind of skip connections, now finally predicting coverage at 32 base pair resolution. So to train the model, we're using both human and mouse data. So we're training the model in sort of alternating batches of human sequence and mouse sequence. And we curated basically one-dimensional coverage profiles to train on. So basically the input to the model is this very large sequence, and the output is a collection of one-dimensional coverage profiles, a very large collection of coverage profiles. And we collected, I think, almost 1,000 human RNA-seq experiments from both the ENCODE and GTEx consortia, which are these amazing resources that have these coverage profiles basically made available for public consumption. We coupled the training of the model with, we trained the model to jointly also predict various epigenomic coverage assays, which we noticed improved the model's ability to generalize and make better RNA-seq coverage predictions, because it's then learning to rely on more salient features. And then we also, of course, included cage data measuring expression at the 5' ends of genes from the Phantom consortium. So after having trained the model on human and mouse sequences, we can now apply it to predict RNA-seq coverage in held out sequences of the genome that the model didn't see during training. And qualitatively, you can see that the model can quite accurately, identify the exons and place coverage over those exons, roughly on par with, with what the measurements look like. And now if you were to sum the total amount of predicted coverage over the exons of the predicted track for a given gene, we can derive a single, scalar gene expression prediction. And if we compare predicted to measured gene expression values, we actually observe quite good concordance on held out test genes across these various sequencing experiments that we trained to predict. So what's nice with these kinds of machine learning models is that they're not necessarily just a black box predictor, but now when we can make these RNA-seq coverage predictions, we can apply various interpretation techniques to those predictions in order to elucidate what features in the original, huge input sequence actually contributed to the RNA-seq prediction. So to illustrate that, what we did here is that we centered the borsoi, half a megabase input window on the liver-specific CFHR2 gene, which is known to be liver-specific. And we then predicted RNA-seq coverage in 5 distinct tissues, including liver and a few other off-target tissues. We then, what we do then is we sum the total coverage over the exons of the CFHR2 gene, so we get like a scalar gene expression prediction per tissue. We then apply this interpretation technique where we rely on the differentiability of the neural network model, and we backpropagate the gradient of the RNA-seq coverage prediction per tissue down all the way to the sequence level so that we estimate the gradient of the prediction with respect to the sequence. So you can think of that sort of as a sensitivity map that tells you how much the RNA-seq coverage prediction in that tissue is perturbed given a slight perturbation to the input sequence. And if we do that, what you see in the upper figure there is basically the most salient region that occurs a bit outside of the CFHR2 gene. And what we find is basically all these well-known liver-specific transcription factor motifs that are effectively predicted to drive the expression of this gene. Now, if you repeat that exercise for many different genes by recomputing the input gradients for many different genes, and you cluster these gradient scores with a de novo motif discovery tool called TF-MODISCO, we recapitulate many known transcription factor binding motifs. And one thing that's quite nice is that if you look at the motifs that are known to be tissue specific, the model really only places high importance on those motifs in the original input sequence when making predictions for the corresponding tissue. So the model is sort of relying on these sequence features in a tissue-specific way. Another really nice thing, now when you have a parametric model of RNA-seq coverage prediction, is that you can use this model to simulate the effect of, in theory, any sequence that you input to the model, of course to varying degrees of accuracy perhaps. But now we can use this as a tool to actually simulate the impact of genetic variation in isolation. So to illustrate that here, I'm showing you a particular gene that has a fine-mapped causal EQTL SNP. It's kind of a deep intronic SNP that occurs at the center of this gene. It's kind of marked by the black star. And then I'm showing you 2 different RNA-seq coverage predictions. So these are coverage predictions made by the borsoi model in whole blood. In blue, I'm showing you the prediction for the reference sequence. And in sort of overlaid in red is the predicted RNA-seq coverage for the mutated sequence. So as you can see, the borsoi model predicts higher coverage almost uniformly across the exons, meaning that the model thinks this is sort of a gain of expression SNP. And now if you actually go out and curate and aggregate RNA-seq coverage in individuals from GTEx that either have or don't have this mutation, we recapitulate the same trend, meaning that the borsoi model could quite accurately predict the effect of this SNP. And then, of course, we can again now apply an interpretation technique, in this case, in silico saturation mutagenesis, where we just systematically mutate every nucleotide surrounding the mutation, and we can then get a picture of what this mutation is potentially creating or destroying at the sequence level. And in this case, this mutation is creating a transcription factor binding motif that is predicted to work in synergy with nearby previously sort of dormant regulatory motifs that now together kind of create this strong enhancer element, and that's what's driving expression here. And so going beyond just one single example, if you then create a set of positive GTEx, fine-mapped causal eQTL SNPs, and you task the model with distinguishing between, fine-mapped causal eQTLs versus a matched set of neutral SNPs. The Borsa model could basically distinguish between those two sets based on its prediction more accurately than the previous state-of-the-art models could. Now, of course, very recently, I think DeepMind came out with this new model called AlphaGenome that seems to improve upon this even further, so that's very exciting. But then another thing that I want to emphasize about modeling raw RNA-seq coverage patterns as opposed to scalar gene expression predictions is that it gives us the opportunity to reason about a genetic variant not just in terms of how much it affects the total gene expression amount, but also how the relative coverage profile may change due to a mutation, allowing us to actually reason about how a mutation may impact isoform processing and other events. So to illustrate that here, I'm showing you a fine-mapped causal polyadenylation QTL, highlighted in black, that occurs sort of in this very long 3' UTR of this gene. And this SNP is basically estimated to have an impact on 3' processing and polyadenylation, basically increasing cleavage efficiency. And the Borsa model at the top coverage plot predicts an increase in coverage upstream of the mutation and a decrease in coverage, as you can see, because the blue is higher than the red, downstream of the mutation, meaning that the model basically thinks that this mutation is indeed increasing cleavage efficiency. And if you look at the bottom plot, coverage plot, that's basically recapitulated in individuals that have or don't have this mutation. Finally, if we apply an interpretation technique, but now instead of checking the sensitivity with respect to just total gene expression amount, we're now computing the interpretation, the ISM, with respect to the ratio of coverage upstream versus downstream of the mutation. And we can see that now we're getting in the sequence, we're getting an interpretation that highlights a CSTF binding motif, which is sort of a canonical polyadenylation efficiency element. And yeah, if you take the Borsa model and you task it with distinguishing between fine-mapped causal poly-AQTLs versus matched negative SNPs, the model is outperforming the previous state-of-the-art models at that task. And with a similar reasoning about how the predicted relative RNA-seq shape changes, you can reason about splice-altering variants as well, quite accurately, but I won't have time to go into that here. Okay, so now I want to highlight some of our more recent work where we're extending this modeling framework to predict cell-type specific expression profiles. So yeah, the motivation is very, very simple. The Borsa model was trained on all these diverse collections of RNA-seq experiments in tissues, as well as cell lines from ENCODE and from GTEx. But it was not trained on any cell type specific data, at least not in terms of primary cell types, which is of course a big limitation, seeing as I said that we wanted to use this model to interpret GWAS studies, but as we all know, many GWAS loci are thought to have cell type specific effects, and so that's a big problem. So to remedy that, we went out and we curated, downloaded many different publicly accessible single-cell RNA-seq data sets and generated basically big tracks of pseudobulk coverage for distinct cell clusters based on the original annotations from these studies. So we downloaded single-cell RNA-seq data from Tabula sapiens, from Tabula muris and Tabula muris sinis, as well as an adult brain atlas from the big consortium. And we now train this new model that we call Borsoi Prime. We call it Borsoi Prime. Well, the prime word comes as a nod to the fact that all of these single-cell RNA-seq data sets are generated with a 3' end sequencing protocol. So you're getting coverage pileups now at the site of polyadenylation. So that's where that name comes from. Architecturally, the model is very similar to the original Borsa model. But now with the big distinction that we're training the model now to predict single-cell RNA-seq pseudobulk coverage tracks for 850 distinct human and mouse cell clusters. So after having trained the Borsoi Prime model, we can now use it to predict RNA-seq coverage at a cell type resolution. So here I'm showing you predicted coverage in microglia, as well as 2 other sort of off-target cell types for the CIBB gene, which was kind of a gene in the held out portion of the data that we didn't train on. And as you can see, the model is correctly predicting high coverage in microglia, but low coverage in other cell types. And if we aggregate total coverage in the predicted bins overlapping exons of individual genes, we can derive scalar gene expression values. And for held out genes, these predicted cell type specific predictions are matching up quite nicely with the measurements. And then finally, of course, again, if we apply interpretation techniques such as input gradients, and we apply them to these cell type specific predictions, we now end up with cell type specific input gradients that we can cluster with TF Modisco and recover motifs and drivers for individual cell types. Like for microglia, we're finding SPI1 and SEBP motifs, for example. Okay, and this is probably the final slide, I think. So the final power here now comes in the fact that we can interpret and reason about genetic variation at a cell type level. So to illustrate that, here I'm showing you the predictions in microglia for a particular SNP that is fine mapped and causal specifically in microglia. And the interpretation of this SNP highlights this sort of weak ETS-like motif that seems to increase expression of this gene. So beyond one example, we benchmarked the model on a few different EQTL data sets, fine-mapped EQTL data sets at cell type resolution, including 2 smaller studies with EQTLs estimated in microglia and in neurons, and then a larger single-cell EQTL data set from the 1K1K cohort as well in PBMCs. I think maybe one plot that I want to maybe emphasize is the scatter plot for the microglia data set to the bottom left. There. So as you can see, on the x-axis, you have the predicted variant effects of these fine-mapped causal EQTL SNPs with a high PIP. And on the y-axis, you have the estimated beta coefficients from the fine mapping. And it's true that the sample size here is quite small, but still the model is the variant effect scores derived from the model predicted tracks are quite concordant with the estimated beta coefficients. Now, crucially, if you look at the bar chart, if you look at the bar chart to the right, you're seeing that actually it's only the variant effect scores that are derived from the microglia predicted tracks that are concordant with the estimated beta coefficients in this cell type. If you look at variant effect scores derived from some other brain cell type and some other predicted track, the variant effect scores have basically 0 concordance, which kind of emphasizes that the model is now truly sort of making cell type specific predictions of variant effects. Okay, so in summary, the Borsa model learns to predict several different layers of gene regulation from DNA sequence, and the Borsa Prime model sort of extends these capabilities by learning to predict sort of cell type specific suitable single cell RNA-seq coverage profiles. And I just want to end by highlighting some other work from our group. For example, Divyanshi's method for including these Borsa variant effect predictions to improve functionally informed genetic fine mapping, or Hans' work on transfer learning approaches for actually fine-tuning the Borsa model towards your own experimental data sets to get sort of a custom model for a particular new data set. And we also recently included more post-transcriptional regulatory assays into this modeling framework. For example, we now have the capability of predicting RNA-seq half-life time course experiments, like transcriptional shut-off experiments, in this sort of profile prediction framework, which allows us to sort of estimate mRNA decay rates and those effects in terms of genetic variants. Okay, so that's all. So thank you for listening, and I'm happy to take questions.
Thank you very much, Johannes, for a fantastic talk. We have time for a few questions with Melina. Do you want to start?
Really beautiful talk, Johannes. Thank you so much for sharing. There's a lot to talk about. I wonder about one question, I wanted to seek your input, particularly in the context of looking at the benchmarking against the fine-mapped EQTLs, where the difference between EQTLs and GWAS fine-mapping or GWAS variants is that the EQTLs are typically much larger in effect size compared to actual GWAS-detected variants that have more common subtle effects.
Yeah.
The examples that, so just to back up, when in our work, when we retrained Bassett back then, or used Informer and or Borzoi more recently, you know, they seem to line up super well with CRISPR validations for large effect variants and less so for subtle effect size variants. So
Yeah.
I wonder, are you thinking about this, and what are your 5 cents?
Yeah, I think that's a great point. And that's also very much what we see in that, you know, when the model is making very confident high effect predictions, the sign of the prediction is almost always correct, and it's really in these small effect size variants where there seems to be a lot of wobble in the interpretations. And you know, I think one really big thing that we noticed, maybe going from Informer to the Borzoi model, was that there's tremendous amount of epistemic uncertainty in these models. You know, there's only 20,000 protein coding genes to learn these relationships from, and so from one model replicate to another model replicate trained on the same data but just different random initializations, you know, some motifs can even flip direction, right? Because the model is uncertain about the connection. And so I think ensembling these models more and more, going from, you know, Informer to Borzoi, we've now, you know, ensembled the model with 4 different replicate models. And I think AlphaGenome now is ensembling up to 50 model replicates or something like that. And they're clearly getting improved variant effect concordance by that. So that seems to be one approach forward. I think ultimately we're still kind of data limited, and we need more training sequences.
Great. Brad.
Oh, that was a terrific talk. Thank you. I kind of have 2 related questions. The first is, what kind of data do you think is most, you know, powers your model the best? What do you want? Do you want knockouts? Do you want sequence variation? Do you want other data types? And, you know, what's needed for your next generation of models? And the second question is, how important was the metadata, knowing, you know, exactly the cell type, the conditions it was grown in, and all this other stuff? And do you think the importance of metadata will decrease over time when your AI models can sort of impute it?
Yeah, that's a great question. Regarding the first question, so we observed a really, really big gain in performance for a number of tasks, but in particular for variant effect scoring, when we jointly trained the model to predict both RNA-seq coverage and DNA or attack-seq coverage in matched tissues or cell types, probably because you're kind of pushing the model to look towards enhanced regions and other salient features to better generalized RNA-seq coverage predictions. So, like, yeah, adding more modalities and more assays definitely seem to be an approach for improving the model further. I'm really not sure at some point I feel like there's gonna be a saturation effect. We're probably covering many cell states already in the, you know, training data that we have. But yeah, like individual perturbations of various, you know, transcription factors, or like a genome-wide, you know, perturbation data set that could probably be great for teasing apart, you know, you can imagine, you know, you have one coverage profile that really shows you what gene expression should be if you knock out each individual transcription factor. That could help the model link that observation down to each regulatory motif more stringently. I think another axis where we really need to grow more is just we need more unique sequences to train on to make these models more accurate. People have been exploring, you know, personal custom genome fine-tuning now with limited success. There's also this big trend now in self-supervised learning of many different genomes from other, you know, related species that could help us improve these models further. I think that was the answer to question 1, and then did you have a second question?
Just about where do you think, how important is metadata going forward?
Right. I think metadata is important for the tracks that we ultimately want to use when scoring genetic variants and interpreting them. But like in terms of adding more data for the sake of improving model quality, in terms of, you know, adding all this attack data over here, even though maybe we don't have perfect annotations for it or perfect metadata, it might still have positive interactions in making us do better for predicting RNA-seq coverage if we can rely on correlations from that attack data. So I think it's a mixed bag.
Great. We have 2 final questions.
So I have two questions. So first question, did you use your model to test the known disease-causing mutation to see whether your predictions are consistent with the known effects? The second question is, did you let's say compare your model to alpha genome to see if your model works better than alpha genome or is it comparable?
Yeah, good questions. Regarding the first question, we have started applying and using the model for specific use cases related to genetic disease. In terms of benchmarking, we've relied mostly on EQTL studies so far, just because those are typically great resources, and you can set up these classification tasks and comparisons against beta coefficients to really quite well. You have a pretty good benchmark capability there. Regarding the second question, we haven't yet. The Borsa model came out a couple of years ago, and Alpha Genome came out a few months ago. So we haven't had a chance to use the Alpha Genome model yet. It's also, we're a company, so we're not allowed to just yet use that model. Yeah.
Great. Sorry, Liz, I overlooked. We had a question up there. Maybe you will need to take your question in the break, actually.
Hey, I'm here.
Hello.
So a quick question on this, similar to the more data approach. There is also a parallel line of models being developed recently, shorter context, single modality from Anshu's lab, and also DeepStar, MPRA screens. Do you think these two approaches, short context, long context, and MPRA, will they be unified in the future in a single model?
Yeah, I think that's a good question. And I think these shorter sequence models trained either on MPRAs or endogenous assays are really nice models. And I think one thing with this approach of doing kind of massive multitask learning, like with Borsoi or Informer or Alpha Genome, is that it's computationally efficient in the sense that you can score genetic variation with regard to many different outputs in one forward pass, which makes them quite nice when you're wanting to investigate some genetic data set where you're broadly interested in variant effect scores across many different types of cell states, which may be harder to do with models that are strictly single task. On the other hand, those models are way more efficient in terms of having a shorter sequence window, so each prediction on its own is computationally retractable. In terms of combining these data sets, maybe. I mean, if you trust that MPRA measurements work the same way in an endogenous context, it would make sense to try to, say, fine-tune these models on MPRA measurements. I think very recently there's been some really cool data sets, new technologies coming out where it's MPRAs, but it's actually in the endogenous genome, like the BigIn platform and dark matter DNA, where they're actually doing these really large rearrangements of the genome and measuring that out synthetically and in reasonably high throughput. And I think those types of data sets are definitely worth, thinking about how to combine into here.
Thank you.
Great, thank you. I think our brains are ready for a break, so please join me in thanking all speakers from those two great sessions.